Hello everyone, welcome to Antibody Engineering and Therapeutics Digital Week, brought to you by the producers of the face-to-face antibody engineering conferences visiting Amsterdam and San Diego later this year. My name is Carter Anderson. I'll be your host for today's session titled New Ways to Boost Recombinant Protein and Antibody Yields in Transient Systems. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you're experiencing other technical issues, hit the question mark button to receive assistance. Anytime during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. All sessions this week are being recorded. You'll be emailed in 24 hours when they're available for viewing. Let's now begin by introducing today's speaker, Dr. Brady Wu, Associate Director at GenScript USA. Thank you for joining us today, Dr. Wu, and thank you to GenScript for sponsoring the event. Now, we'll hand it over to you.
Thank you for your nice introduction, Carter. This is Brady Wu, Associate Director of GenScript. I'm in charge of the protein production. It's my great pleasure to join this webinar to introduce the new systems we are working on to boost the recombinant protein and antibody yields in transient systems. So the talk will be divided into three parts. The first one is a general introduction of GenScript and our services. And the second part will be the efforts by GenScript on the improvement of the transient systems. The third part will be efforts by GenScript on immunogen production specific protein. So GenScript is a global Bio-CRO, which was founded in 2002. Right now, we are publicly traded in the Hong Kong stock market. We have over 1,700 employees worldwide. We are a top company globally for gene synthesis. But more than that, we are a top Bio-CRO in China. We are providing one-stop service, and we are specialized in biologics discovery and development. So we have a global headquarters in Piscataway, New Jersey, and we have a main production site in Nanjing, and we also have an office in Japan and Europe. So today, my focus will be some part in drug discovery and development. So basically, we provide one-stop solution from target to R&D, starting from target assessment and validation, antibody lead generation, optimization, and the development parts such as cell line development and GMP compliant manufacturing. We had a lot of experience in terms of target type. Well, we have worked with a single transmembrane target, multiple transmembrane target, and soluble protein targets. In terms of indications, we have worked with indications such as cancer, pain, diabetes, and autoimmune diseases. So we have more than 400 discovery projects in total, and about 25 CMC projects. Right now, we have one project going on in the clinical trial. So we truly have one-stop biologics platform from discovery to development. That was divided in several parts, such as antigen selection, biophysical assay, biochemical assay, functional assay, such as internalization assay, neutralization assay, immune cell assay. So today, my focus will be recombinant protein antibody production and protein production used in drug discovery and development. So firstly, I want to simply introduce what is transient production. So in the contrast to stable production, we have a transient system which you prepare cell and DNA separately, and you mix DNA with transfection reagent, and then you add the DNA to the cell culture. So that's really straightforward. We don't have screening phase, and we don't do clone selection. So in the industry, we have three different systems, transient, stable pool, and stable cell line. They have pros and cons for transient systems. They are the fastest time to material. The material means you have some reagents for drug discovery and some reagents for reagent use, such as recombinant antibody used in the assay. So the major applications for transient are reagents and early drug discovery antibodies. For the stable pool, that means you do a stably transfection, but you don't select the single clone. So normally that will give you a higher titer than transient, but the pool will decay during repeated production. So stable pool was normally used for large quantity materials such as gram level materials, and also because stable pool is on the way to the cell line, you can generate a cell line while generating materials. So for the stable cell line, we use that for stable production or commercial production, but it has a very long setup time from 3 months to 5 months. So the major application is industrial production. So the major con of transient production is moderate titer, but GenScript has put a lot of efforts to improve the titer and push the boundary of transient production. How we are going to do that? So there are a lot of methods to tune in the transient production. If you look at the cell pathway, actually the cell we are using is no different from the cell in stable production. So we are looking at different pathways in the cell lifetime, such as apoptosis, cell proliferation and growth, protein folding, and cell metabolism. All the factors could contribute to the cell death and the cell leaving, and also their capability to produce proteins. So we want to do something on this matter. So the first thing we do is to produce a new anti-apoptosis system, which we tune the cells to keep it live longer. Let me explain this figure. So we use a normal system called 293 and a new system which we have tuned the apoptosis of the cell, which I called 293G. So from this figure, we can see 293G has better viable cells and viability compared to the normal system measured by this figure. So we tried 20 different constructs in the cell line and see how they behaved from day 3 and day 6. And so normally we do transient production in 6 days. So in both cases, day 3 and day 6, 293G has better viable cells and viability. But we don't know yet whether that can be transformed to better titer. So in this figure, we can see with some low expression antibodies from human and mouse, in total of 12. In all 12 antibodies, this system improved the titer of the antibodies compared to the regular system. So for the human antibodies, we improved the titer by 3.5 to 9-fold. And for the mouse antibodies, we improved the fold from 4-fold to 5.5-fold. So that system works for low expression antibodies. How about high expression antibodies? So we tried another dozens of antibodies. So for our system 293G improved the human and rabbit antibodies titer by more than 10-fold. And for the 3 more mouse antibodies, we can improve titer by 2 to 6-fold. Again, we show this new system can improve the titer universally, but that's for monoclonal antibodies. How about some other more difficult antibodies? Just show a figure of the basic antibodies we have done. So in these 2 different systems, in 100 milliliter culture media, we can improve the titer by 5-fold compared to the regular systems. So that's very nice of that. So we already have a system which can improve the titer from 4-folds to 10-folds just by touching the apoptosis. How about some other methods we can use for the improvement on this system? On this 293G system, first we try the cell growth regulator. So the figure has a lot of contents. So we try to control just this system, and we try 2 different cell growth regulators as 2 different components. you can see from this figure, component 2 alone can improve the expression further by 85%. But the component 1 does not work alone. So we'll carry on with component 2. How about some other system we have touched based on the metabolic pathway optimization? We try 4 more components, and for components 4, 5, and 6, we show improvement on the titer. For the component 3, it does not work alone. So we add all of them as a 25% increase. So basically, we put 25% of the component and 75% of the antibody gene in the system. So the last one is a complicated DOE system we are trying. So it's a design of experiment. Let me explain this figure. So we tried 28 different combinations of all 4 components we have tried, 2, 4, 5, and 6. So these 4 components we tried worked in the previous single components experiment. So we see, how about we try 4 together? So we tried a lot of different ratios, and we found the best ratio is 10% component 2, 4, and 5, and 20% of component 6. So they are cell circle regulator, protein folding enhancer, growth stress releaser, and energy booster. So combination 18 showed the largest improvement. The titer was nearly doubled compared to our regular 293G system. It was proved to be an effective cocktail. So we'll probably incorporate that in our standard experiment. And so right now we have a nearly tenfold increase on titer compared to a regular system without any tuning on this system. So right now, I hope I convince you that GenScript has the capability to build up a good transient system. And with this system, we also need to do some process control with state-of-the-art instruments. We have info shaker, a high throughput shaker, which has very good control on the humidity and oxygen level and the shaking speed. And we have white cell, which can counter cell very accurately, automatically. And we have Tican, which can purify the antibody automatically. So we have very good quality control during our process. So let me revisit the target from IND. So in the target assessment and validation part, normally we will need immunogen production. Compared to, there are several ways to do the immunogen selection, such as DNA immunization, VLP cell line, protein immunogen, and peptides. Protein immunogen is the most common one because you can provide active proteins such as active ECDs and secretory targets. And active proteins will give you active antibodies. For other methods, because you cannot get very pure antigen from DNA immunization, VLP or cell line, protein is normally the best case for your antigen. But it's not trivial to get proteins. So antibody, we consider that as one type of protein, and we also have other type of proteins. So compared to antibodies, protein has multiple subcellular locations, but antibody can only be secreted, and the protein has complicated modification, but antibody has simple glycosylations. For antibodies, we have very neat protein A or protein D column to do simple purification. But for proteins, the purification can be complicated. And also the expression level of antibodies are quite predictable. Depends on the types of protein, the titer of proteins are often low, especially compared to the recombinant antibodies. The proteins can be monomer or oligomers. So that really decides whether the protein can be functional. But for antibodies, it's always for monoclonal antibodies, it's always 2 heavy chains plus 2 light chains. So when we are doing a recombinant protein production, we have to consider a lot of factors such as cytocellular location of the native protein, host cell systems, cell homeostasis, tags fusion partners, and chaperones and purification schemes. GenScript has a lot of expertise on recombinant protein productions. We have tried FC fusion proteins, cytokines, enzymes, glycoproteins, intracellular proteins. So if say you compare to antibodies, there are a lot of different formats of proteins. And normally antibody will give you very high purity, either by SGS-PH or HPLC, but not so far for protein. For example, when we are trying this protein, it's a TGF-beta family protein from left, TGF-beta 1. This protein needs a very good fusion partner in order to get an active and pure protein. Similar for the full-length human AMH, without a fusion partner, you cannot get the proteins. So for this type of protein, the fusion partner is very important. We have some other cases. So this protein was with a his tag. So normally nickel column is very good for the his tag, but when the protein was at very low titer, it was mixed with a lot of impurity other proteins. So the specificity of the nickel column was not enough to get a high purity from one-step purification. So we use a neat method to use antibody affinity column. So we use anti-his antibody to get purified protein from this case. So this protein has some other issue. It has aggregation issue with a small tag, and in protein extraction and purification, the tag selection could be very important. So we did some optimization by changing tags. By changing from a small tag to a larger tag, we have an extra shield to screen out the proteins during expressions, so we can get high yield and purity protein with the changing tag method. So this protein is another interesting case. It was degraded a lot just during expressions. You can see for the non-reducing gel, the protein already showed a lighter status degradation, which will make the purification impossible. So we change the way to culture the cells. So we monitor the cell viability and the density very accurately, and keep the cells from the start to over at a very high variability. So the cells will not release some enzymes which will lead to the protein degradation. By this method, we can improve the protein purity during expression. So right now, I'm touching based on a favorable strategy by a lot of protein experts. So we use a P2A fusion peptide to link two proteins, because sometimes we want to get a protein complex, especially heterodimer. So with this strategy, we can make sure that the protein is expressed as a 1-to-1 ratio, and we just use one plasmid in the expression, and they are quite close in space during expression. That will make sure that you get a functional heterodimer. So there are a lot of tricks in protein production in mammalian cells, but in any case, there are some proteins cannot be done in mammalian cells. For example, we have done a lot of difficult member proteins such as Claudin 18.2 and CXCR4 or GPCR. GenScript has a lot of different systems, so we try that in Baculovirus. So the Baculovirus system has larger cell surface, so that means it can contain more membrane proteins. And when we did a little bit trick on the protein purification part, we got very high purity proteins which can be used as protein antigen. So right now, I hope I have convinced you that GenScript has developed a novel system and which can improve the titer of recombinant antibody and proteins by about 4-fold. And in the future, we hope we can improve the titer of recombinant antibody and proteins by 10-fold. And we have developed scientific and technical expertise doing recombinant proteins for drug discovery and development purpose. In the future, GenScript will continue to improve the capability of our service and products. We'll keep our focus on reagent and drug discovery and development. With your help, we will all make research easy. So our slogan is make research easy, and I think that's more important to make research easy for the people. So that's last slide of my presentation. And also we have a new promotion for the recombinant antibody services. You will receive 10% off for recombinant antibody service with the following codes: RAB 10%. With that, I'll hand over the webinar to Carter.
Thank you, Dr. Wu, for an excellent presentation. We've received a few questions already, but we'll give the rest of you a moment to enter questions in the Q&A box to the left of the slides. Before we begin the Q&A, I'll run through some quick announcements. First, I'd like to again thank GenScript for sponsoring Digital Week. Next, I'd like to quickly draw your attention to our face-to-face antibody engineering and therapeutic events visiting Amsterdam, June 5th to the 7th, and San Diego, December 9th to the 13th. At these market-leading annual events, you can learn from experts like Dr. Wu and network with hundreds of your peers. With that being said, let's now begin the Q&A. Dr. Wu, are you all ready?
Yep.
Great. All right, we have a first question here. Do you try different tag placements such as the 5' end or 3' end or in between?
Yeah, that's a very good question. Normally, we will try the tags at the end of the protein, either 5' or 3', or you can call N-terminus or C-terminus. So for mammalian, we will try to segregate or produce our proteins. So normally, we will try insert C-terminus or 3', but sometimes N-terminus or 5' will also work. If I prefer, I will prefer the 3'.
Great. All right. We have a second question here. Does this new system provide higher yields for difficult proteins such as membrane proteins or toxic proteins, and if so, how?
Yeah, that's a very good question, but sorry, we don't have time to look at it yet. In order to get a very high yield on membrane protein, you have to get very high density because unlike segregated proteins, the surface of the cell will determine the yield of a membrane protein, and the cell number will determine how big of a surface you can get in certain volume of the culture. So if we can culture the cells we have to a higher density, I think that will be a good system for membrane protein. For now, we don't know yet, but we will try that in the future.
Okay, thank you. We have a third question here. Why do you think that some mammalian systems prefer certain protein tags?
Yeah, actually, it's a very good question. Well, based on our experience, mammalian system has very good expression performance if we fuse a protein with FC tag. You know, in a living system such as human or mouse, a protein with FC tag or antibody will have a longer half-life. My guess is that it is related to the longer half-life of FC fusion proteins is related to the high titer when we are using single clones to produce our proteins. I think it's related. That's a story for FC tag. But for some other small tags, flag tag is better than his tag because the proteins produced by mammalian system will have a lot of impurities which can compete with compete with your target protein on the nickel column. So for small tags, I think anti-flag tag will be a better option.
Thanks, Dr. Wu. That's all the time we have for questions today. If your question wasn't addressed, the speaker will reach out to you directly. Be sure to download the presentation slides in the resource box, and we'll send you a reminder when the recordings becomes available. Before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Connect 365 Life Sciences, have a great day.